So let's start off by making a list. What are the things that we should be thinking about in a course that covers plant physiology? What are some of the basic issues, let's say, that plants have to deal with in their day-to-day existence?
Obtaining nutrients.
Nutrients. Getting nutrients from the environment, but not just that, because those nutrients, if we take up nitrogen from the soil, it doesn't stay as nitrate or ammonia in the plant. They also get assimilated, right? So we've got assimilation of the nutrients as well. Yes, somebody had their hand up here. Yeah. Yeah, water. Water's a big thing. What else? Homeostasis, what do you mean by that?
Maintaining a livable temperature.
Plants keep a constant temperature?
Well, maybe not.
No, they don't keep a constant temperature for sure. Do they keep a constant bathing fluid inside the plant? In other words, we have blood and lymph circulating around our body. Do plants have something similar to that they maintain? As long as you don't answer it, that's okay. So there is some level of homeostasis. It's basically maintaining the internal environment of the plant within certain constraints. Temperature largely doesn't happen in plants, but for example, osmotic characteristics, ion composition is highly regulated in plants. Yes, back here.
And light interception.
Light, yeah, light's a big thing, right? And we need to think about it both in terms of competition for light and how that light is actually utilized. What else?
Growth.
Growth. So what about growth?
Well,
Why do you say growth? Everything's got to grow.
Yeah, well, I mean, cell division and then cell elongation.
In what context is there something unique about this in plants? There's aspects of growth that are completely unique to plants. Yeah?
Is it mostly like they replicate really fast in the beginning and then they grow longer based on like adding more water to the cell or something?
Yeah, so cells divide quickly and then enlarge and specialize. But that's actually something that's common to, well, enlargement is much more in plant cells in general than it is in animal cells. But that's fairly common. I'm thinking about, for example, well, go ahead, what you can say.
Like the apical meristem?
Yeah, so we have meristems. Meristems are basically post-embryonic growth. All new cells come from the meristems, whether they're shoot or root meristems or whether they're lateral meristems in woody plants. Okay, so that's really different. But there's another aspect of plant growth that's different. Yeah. Nope, right, I'll learn your names, so I'll get your pictures. Say that again?
Is it the haploid diploid cycle?
Oh yeah, so there's an alternation of generations associated with, let's put that down here, and this related to reproduction. What I'm getting at here, the thing I'm missing, is the role that cell walls play. Cell walls both constrain the way the plant grows, but also dictate the way the plant grows. That is, how big a cell gets, what its shape is determined by the enlargement of the cell wall. It's not a random process. It's very highly constrained, and obviously quite different than what happens in animal cells, where there's no cell wall that constrains growth. Okay? What else? Yeah?
They have to defend themselves.
Yeah, defense. You know, so when the bear walks in through the door here, we can jump out the windows, right? We can escape. Plants can't do that. They're stationary. They're sedentary, right? And so they have developed a whole range of mechanisms of dealing with things that eat the plants, things that want to take goodies out of the plant cells, parasites and things like that. So plant defense is a big component of this. And what this basically represents, as far as what plants do, we refer to as secondary metabolism. plants make a whole bunch of specific compounds that are related to defense, related to preventing or limiting things from eating the plants, things from invading the plants and giving the plant diseases and things like that. The secondary metabolites or secondary metabolism distinguishes it from the sort of thing, the metabolism that's common to all organisms. And we should put that on the list down here. Primary metabolism. So photosynthesis produces sugars. Nutrient acquisition takes up phosphorus and nitrogen and stuff like that from the soil. But those got to be converted into proteins and nucleic acids and lipids and all that sort of stuff. That's the stuff of primary metabolism. Okay, so we got a pretty good list here. If you compare this list with the chapters in the textbook or the list of lecture topics, we pretty much got it covered. Right? These are the sorts of things that we're going to be talking about. I think the one thing that we don't have on the list is environmental response. In a sense, defense is a type of environmental response to biotic factors. But we also need to include environmental response to abiotic factors, changes in temperature, changes in water availability, nutrient availability, and things like that. Okay, so by the end of the course, you should be able to talk about how the details that are available to you in the textbook relate to all these different things, and use that information to say, okay, suppose I suggest that a plant is exposed to certain environmental conditions. You should be able to tell me how the plant's what the plant has available to it to help it respond to that. Okay? All right. So let's spend a few minutes talking about some of the things that are unique to plant cells. If we think about plant cells, they got a lot in common with all eukaryotic organisms. They got nucleus and they got cytoplasm and all that kind of stuff. But there's some things that plant cells have that are unique, that put a whole new perspective on the way that plant cells do things. And one of them is a plant cell wall. So we'll have a whole lecture topic where we talk about plant cell walls, and then we'll talk about them again in terms of the role that walls play in relation to development and growth. Because as I said, the walls, the enlargement of the wall is what dictates how the cell volume and the cell shape changes. So plant cell walls put very important constraints on plant cells. One, plant cells can't move. It's a big difference in development between plant and animal cells. In animal cells, when two cells divide, there's nothing that says one of those two daughter cells can't migrate halfway across the body to become some new type of cell. That doesn't happen in plants. When a cell divides, those two cells are locked in position relative to each other. That puts a lot of constraints on development. And we'll talk in detail about this when we talk about development in plants. Plant cells also play a huge role in water relations. We cannot tolerate in our body changing the internal osmotic conditions of the fluid that bathes our cells. All right? Because animal cells don't have a rigid wall, the fluid that surrounds them must be isotonic, must have the same osmotic characteristics as the cytoplasm. Not true in plants. The thing that bathes the cells in plants is basically dilute water solution. Right? And the reason the plants can withstand that is because water wants to move in osmotically. We'll talk about this in the next couple of lectures. But the cell wall resists that. Right? So cell walls play a very important role in distinguishing what plant cells are capable of doing compared to the average cell in a bacteria, a yeast, or an animal. Plant cells contain plastids. So when we think about plastids, we think primarily about chloroplasts. But chloroplasts are just one of a group of related organelles called plastids. So we have chloroplasts. There's a group of plastids called Leuco Plas. Leuco Plas means they're not colored at all. The most common type of leucoplast is an amyloplast. We'll see that these are involved in starch metabolism. And there are colored plastids that are involved in other things besides photosynthesis. We call these Chroma Blasts. You're familiar with these, like tomatoes. The red of tomatoes are chloroplasts that have turned red by accumulating the carotenoid lycopene in them. Right? So these are also found in the petals of colors of petals and flowers and things like that. So these are all developmentally related to each other from a precursor called proplastids. They can develop into any of these things depending upon the cellular conditions. Proplastids are inherited through sexual reproduction, usually only in the egg, sometimes in the sperm. So it means, because we know that chloroplasts and actually all of these plastids are semi-autonomous organelles. They contain genes that encode a small fraction of the proteins that function in chloroplasts, that we have non-Mendelian inheritance of traits associated with plastids. Because in many plants, the plastids are only inherited from the maternal parent through the egg. Sperm is too small to hold the proplastids. Let's see. The other interesting thing that we need to think about, we've already mentioned. The fact that cell division in plants is limited to specific regions. Not true in animals. In plants, it's the meristems, the shoot apical meristem, SAM, the root apical meristem, in all plants accounts for growth lengthwise, upwards and downwards. In woody plants, like trees, there are also lateral meristems that account for increase in diameter of the plant. But all plants, and what we're going to focus on when we talk about them, are the apical meristems at the shoot tip and the root tip. So in the embryo, in the development of plant embryo, lots of cells divide. But once the embryo is formed, once the seed dries down, and the seed imbibes water and starts to grow again, all that post-embryonic growth, every new cell, is the result of cell division in one of those meristems. So the meristems account for all post-embryonic growth and development. It's obviously quite important in determining what's going on in a plant. Okay, so cell walls, plastids, and meristems. Those are the sort of key things that we're going to come back to many times over the course of the semester to think about unique aspects of what plants do. Okay, let's sort of go back to the level of introductory biology for a little bit. So here's a picture of a typical plant cell. Well, maybe not such a typical plant cell, but you should know about all the stuff that's in there. So when we talk about endoplasmic reticulum and nucleus and Golgi and chloroplasts and mitochondria, you should know about all that stuff, right? If you don't, you better go back and read some of it. There's some information in the first chapter of the textbook. But if you're missing basic cell biology, then you need to bone up on it, because it's going to turn out to be important in a lot of different circumstances. So we know that cells are basically the fundamental unit of life. So everything that a plant does is dependent upon processes at the cellular level. That's true for all organisms. But we think about it in an animal, for example, in humans. We have a heart that mechanically pumps to circulate fluids through our body. Plants circulate fluids through their body as well, but there's no mechanical pump. Everything that happens to drive long-distance transport in plants is the result of osmotic processes at the cellular level. Okay, so we're going to spend a fair amount of time this semester talking about processes that are happening at the cellular level and understanding how those give rise to the things that we see at the whole plant level or even at the ecosystem level. So cell biology is going to be important here. So let's talk about some of the things in here that I expect you to know about. So we talked already about the cell wall, and we'll come back to that again at the very end of the lecture. As we move in from the cell wall, what's the next thing that we encounter?
Cell membrane.
Cell membrane. Okay. So what's important about the cell membrane? Why should we spend any time talking about it? How about you in the back with the green and black shirt? Yeah, you. Yeah, so it determines what goes in and out of the cell. How does that happen? What does the plasma membrane, what characteristics does the plasma membrane have that make it, we like to use the term selectively permeable? Some things can go in and some things can't. What are the characteristics of the plasma membrane that allow that to happen? How about you in the gray shirt right back here? Yep, you. How does the plasma membrane, what characteristics of the plasma membrane allow it to be selectively permeable? Yeah, so proteins that allow specific things to go across the membrane. What else? Yeah, the membrane itself, right? So the easy way to think about it, not just for the plasma membrane, but for any of the membranes in a cell, is that the bilayer, for the most part, is the barrier. There are relatively few things that can cross through a lipid bilayer. Small molecules like water and gases, they can go through pretty easily. But things like even simple sugars, glucose, can't pass through a membrane by itself. It needs to have a protein. And those proteins, basically the proteins that are in membranes that function and transport, are enzymes. They're enzymes that are catalyzing the movement of a molecule across the membrane that wouldn't otherwise cross the membrane. Okay? So when we think about the plasma membrane, or we think about any of the other organelles, one of the common themes that's going to be there is selective permeability. So let's think for just a second about, imagine a photosynthetic cell from the leaf of a plant. Let's compare that with a unicellular photosynthetic prokaryote. No chloroplasts, no internal membranes. What are the differences between what that prokaryote does to live and what the plant cell does to live? Are there any big differences?
Well, the prokaryote will take in its food by engulfing other small animals.
It potentially can, yeah, but it's a photosynthetic prokaryote, so presumably it's getting most of its nutrition autotrophically rather than heterotrophically. What you're saying is correct. Even most photosynthetic prokaryotes, even most photosynthetic eukaryotic algae, have the capability of either taking up organic compounds or even engulfing things by phagocytosis. But that's a sort of a separate issue. Are there big differences between what a prokaryotic cell does and what a eukaryotic cell does?
Can't the prokaryotic cell move with like cilia?
Yeah, okay, that's true. The prokaryotic cell can move. Yeah.
Does the photosynthetic processes on a prokaryote happen on the actual cell membrane versus in a membrane?
Yeah, so that's an important distinction. All the membrane-requiring processes, like photosynthetic electron transport, respiratory electron transport, in a prokaryote happen in the only membrane that's there, in the plasma membrane. What I'm trying to get you to think about is, in fact, there are virtually no differences when we think about the breadth of things that cells do. There are very few differences between what a prokaryote and eukaryote do. But there's a lot of structural differences. Why all these organelles? What do the presence of organelles do for eukaryotes that's not possible in prokaryotes? I just told you that virtually everything they do is the same. There's very few processes that a eukaryote can do that a prokaryote can't do. So why have organelles? What benefit does that give? How? Yeah, more efficient, but how?
Plant cells can store the products so they can use them later.
Prokaryotes can store stuff too. They often store things.
Well, there are more like membranes and surfaces where things can happen because there's
Yeah, except for actually in photosynthetic prokaryotes, what you find is the plasma membrane invaginates and folds in, and all those membranes in there are, for example, involved in photosynthesis. So there can be a ton of membrane in a prokaryotic cell.
You can have specializations that can have a part of the cell that's working on one thing that you can't, that has, component that you can't use it.
I'm just looking for English literature.
Yeah. But you can have components that are maybe toxic to other processes, and that way you can separate.
Yeah. So let me give you an example that we just talked about. In the prokaryotes, the electron transport associated with respiration and photosynthesis occur in the same membrane. In eukaryotes, they occur in separate organelles. In prokaryotes, the glycolysis associated with respiration and the Calvin cycle associated with photosynthesis occur in the cytoplasm of the cell. There's a lot of common intermediates there that actually make it harder to regulate those processes. So by the compartmentation, it provides a much cleaner way to regulate processes independently of each other than can be done in eukaryotic cells. So the difference between prokaryotes and eukaryotes in terms of the actual processes that are there are very small. The difference is how those processes are regulated. Compartmentation gives a lot of opportunity for regulation that doesn't happen in prokaryotes. Okay, one aspect of this membrane mess that I want you to be thinking about, there's an emphasis in it on the readings in the first chapter, is related to the endomembrane system. So what are the organelles that are associated with, or what are the membranes that are associated with the endomembrane system? Yup. What else?
Golgi apparatus.
Golgi. What else?
Cell membrane.
Plasma membrane. Yep. What else? Nuclear envelope. Yep. What else?
Vesicles.
Yeah, lots of vesicles. Let's switch the question around. We've named almost everything in there. What isn't part of the endomembrane system?
Cytoplasm.
Well, yeah, the cytoplasm's it's not a membrane, so we're not going to worry about that. But yeah, it's not part of the endomembrane system. What other membrane-containing organelles are not part of the endomembrane system? Nope, vacuole is part of the endomembrane system too. Mitochondria and chloroplasts. Yep. Why? Why are mitochondria and chloroplasts not part of the endomembrane system?
Because they didn't originate from the vacuole.
Yeah. How did they originate?
They were other bacteria or small.
Yeah, so you've heard about the endosymbiotic hypothesis or endosymbiotic theory, that chloroplasts and mitochondria were free-living prokaryotes that were taken up by phagocytosis, but not degraded. The fact that chloroplasts and mitochondria contain their own DNA, their own ribosomes, tells you that they have a history of being a free-living organism. I've actually done a fair amount of work on organisms that were precursors to chloroplasts and how that endosymbiotic event might have happened. And there's just a ton of really interesting stuff. We could spend a whole semester talking about that. So you're correct that the things in the endomembrane system are ultimately derived from invaginations of the plasma membrane. Invaginations that can form individual vesicles, that can fuse together to form ER, Golgi, nuclear envelopes, all those sorts of things. Chloroplasts and mitochondria are separate. They came in, they were taken up in one of these vesicles and were retained. We'll see that they lost most of their genetic material, but they retain a little bit of it. Okay, so why do we care about this? Why should we worry about the complexities of membrane movement associated with the endomembrane system. What is this important for in plant cells?
Everything.
That's pretty good. Let's say most things. One of the things that we talked about already. How does a plant make a cell wall? Much of the material that forms the cell wall is produced in the Golgi and is exported in little vesicles that when those vesicles fuse with the plasma membrane, the contents of the vesicle are released. Basically everything but the cellulose microfibrils is exported from the cell via the endomembrane system. How do you add or remove proteins from the plasma membrane? So they may be transport proteins that are needed because the plant runs out of nitrate, and so you have to put high affinity nitrate transporters in the plasma membrane. Or developmentally, how do you insert or remove specific receptors, protein receptors from the plasma membrane? It's through this system. Right? So the flow of membrane and the proteins that are enclosed in the membrane are very important in all sorts of things that happens in plant physiology. Okay? So having a basic understanding of what's going on here, both in terms of the membrane and in terms of the proteins. Proteins that are going to be secreted are synthesized on ribosomes that are bound on the rough ER as opposed to free in the cytoplasm. Proteins that are going to end up in the membrane of the endomembrane system. So in the nuclear envelope or in the vacuole envelope or in the plasma membrane. Proteins, integral membrane proteins, are made in the rough ER and exported through this system to get them out there. Okay? All the lipids that make up those various membrane systems, except the chloroplasts and the mitochondria, are produced in the ER and spread around the system. So when plant cells grow and divide and differentiate, all of this is dependent upon what's happening in the endomembrane system. Okay, so make sure that that's something that you're comfortable with. I already mentioned the fact that plant cell walls play an important role, and this is just sort of an example of where two different aspects of the plant cell structure become important. When a plant cell divides, so here we have late telophase after the nuclear envelopes of the two daughter cells are starting to form. The next thing that happens is formation of new plasma membrane and cell wall between these two cells. And the way that happens is comes from vesicles that are derived from the Golgi. The vesicles that are derived from the Golgi, these little blue things here represent vesicles that aggregate on the cytoskeleton at the plane of cell division. When those vesicles fuse, the membrane becomes the new plasma membrane of the two daughter cells. And the contents of those vesicles is the glue that holds the cells together. It's called the middle lamella. We'll talk a lot more about this later on in the semester. So the cytoskeleton directs these vesicles to the plane of cell division. And those vesicles fuse, forming simultaneously the two plasma membranes of the daughter cells and the cell wall, the first layer of the cell wall that holds those two cells together. Okay, so here's an example of where the endomembrane system and the cytoskeleton interact together to dictate this particular process. Okay, somebody mentioned vacuoles. Vacuoles are, the role that vacuoles play in plant cells has been understated for a long time. When we look at, this is a micrograph of a leaf mesophyll cell. So we see a nucleus and we see some chloroplast, and it's pretty out of focus. It doesn't really show up very well. But here's the cell wall, and all this volume here is a vacuole. So in one sense, vacuoles are important in plant cells because in many cells they take up most of the volume. One of the other things that distinguishes plant cells and animal cells is that plant cells tend to be much larger than animal cells. And the thing that takes up most of the volume of large plant cells are vacuoles. Why? Why would, if you're going to have a large cell, why would it make sense to have most of the space in that cell taken up by a vacuole? Yeah. Yeah, okay, so that's actually one I hadn't thought of, but that's a good answer. Whatever's in the vacuole is probably cheaper to produce than what's in the cytoplasm. And another thing. What determines, for an animal cell, what determines the maximum size of the cell? What are the physical constraints that limit how big an animal cell can get?
If the cell gets too big, then the cell membrane will break.
No, you can add more membrane and the cell would continue to get bigger.
Rate of diffusion?
Diffusion in what sense?
In the sense of like, if the cell gets too big, then not everything will get what's...
Okay, so you're talking about diffusion within the cell, in the cytoplasm.
Diffusion into the cell.
Or diffusion into the cell. In what sense do either of those affect any processes? Why do we have to worry about diffusion?
Surface area to volume ratio.
What about that?
If you grow larger, the ratio between volume and surface area, like there's less surface area per volume, which means there's less space to have things to go in and out per
And do cells need to have things going in and out? Yeah, they have to exchange mass and energy with their environment to be able to live. Okay, so surface to volume ratio is a very important one. If you have a big cell that's mostly vacuole, the actual volume of the cytoplasm is this little tiny strand around the outside and some stuff up there. The cytoplasm is quite small, but it's got a large surface area, right? So plant cells can be structurally larger by enclosing most of the volume in a vacuole. So does that mean that the vacuole is just empty space?
No.
What's a vacuole do? Give me some examples of what a vacuole does.
Storage,
Stores what?
water,
Water, okay. There's a lot of water in there, yeah.
and
What else?
other stuff.
Other stuff. Yeah, let's be more specific than other stuff. Yeah.
Everything that's somewhat toxic to the cells.
Yeah, so that it can store toxic things in there. A good example, epidermal cells store plant defense compounds. Things that when the cell gets broken open, inhibit, for example, a grasshopper that's eating the leaf, inhibit digestion of the grasshopper. If those compounds were floating around in the cytoplasm, they'd interfere with processes in those cells. By socking them away in the vacuole, the only time they get used is when the cell gets broken open. Right? So it can be used as storage for compounds that would otherwise harm the normal function of the cell. We'll see that vacuoles get used for all sorts of things. So the general idea that the vacuole is a bag of water that takes up a lot of space, which if you took plant physiology 30 years ago, was what most people thought. Vacuoles are a lot more complicated in the things that they do. They play a huge role in regulating the composition of the cytoplasm by transferring ions in and out of the cytoplasm. Okay, we'll talk a lot more detail about that. We talked about plastids already. This is a chloroplast, and we'll spend lots of time talking about photosynthetic aspects of this. And we mentioned that chloroplasts are just one of a larger family of plastids. But here's a picture, fluorescence microscope and electron microscope of colorized electron microscope of plastids, showing some rather weird things that we don't typically see with plastids. What are these long, stringy things sticking off there? These pictures were taken by putting green fluorescent protein, GFP, small soluble protein that fluoresces green, in the stroma, in the liquid enclosed by the chloroplast envelope. So that means the stroma has these long extensions. What are these things? You don't see about them in any textbook. They're actually discovered by a postdoc in Maureen Hansen's lab over in molecular biology and genetics. These things are called stromules. They're long extensions of chloroplasts that actually join to other chloroplasts. Sometimes they even join to other organelles like mitochondria or peroxisomes. What they're doing, we don't have a clue. Well, we're starting to figure it out. So this is an example of the idea that our knowledge in how plants work is continuously advancing. What's going on with these stromules? We don't really know, but clearly they're important. We've demonstrated that you can exchange materials between chloroplasts, between two chloroplasts, or between a chloroplast and a peroxisome. So there's something going on there that's probably important. Okay, so here's a chloroplast, and here's a picture that shows two peroxisomes. Peroxisomes are in this category called microbodies. People used to say that microbodies were unique to plants. We now know that they're not. There are peroxisomes, or things that are involved in dealing with reactive oxygen species, things like hydrogen peroxide, in all eukaryotes. They're just more prevalent in plants. So it's another category of membrane-bound organelles that we need to be thinking about. Let's see. Cytoskeleton. We're going to spend a lot of time talking about various aspects of the cytoskeleton. So here's a picture of, these are palisade cells in the upper layer, the photosynthetic cells in the upper layer of a leaf. They're very elongated, and this has got green fluorescent protein that's labeling the microfilaments that underlie the plasma membrane. So you're seeing a network of microfilaments that we'll see play all sorts of roles in determining cell shape, cell size, cell growth, and things like that. Okay, so cytoskeleton, one of the interesting aspects of cytoskeleton that we'll talk about, and it's important that you keep in mind, is it's highly dynamic. It can take itself apart and rebuild itself in other ways to accomplish specific processes. One of them you're already familiar with, mitosis. Right? The microtubules that are involved in the mitotic spindle, are they there throughout the cell cycle? No. There's microtubules throughout the cell, but when mitosis is ready to happen, those microtubules separate, they break down into their individual parts, reform the mitotic spindle, play the role in separating the chromosomes, and then that mitotic spindle disintegrates and the microtubules reform in other places in the cell. It's a highly dynamic set of things in the cell that are involved in moving things around in the cell, moving organelles around, shaping the cell, dictating how the cell wall is laid down. We'll see lots and lots of examples of this. Okay, I want to finish with just one thing. We talked about cell walls. One of the unique aspects of plant cell walls that we're going to spend a fair amount of time talking about in a lot of different contexts are plasmodesmata. Plasmodesmata are basically cytoplasmic connections between two adjacent cells. Okay, so there's here's a cell wall, here's a cytoplasm of one cell, here's the other, here's the plasmodesmata. There is a lining, a membrane lining. So the plasma membrane of this cell is continuous with the plasma membrane of this cell through the plasmodesmata. There's also a piece of ER. It's called the desmotubule that goes through the plasmodesmata from one cell to the other. And there's literally cytoplasmic space between in the plasmodesmata. We'll see that plasmodesmata play hugely important roles in communication between cells, whether it's communication involving metabolic intermediates in different pathways. So for example, in C4 photosynthesis, we'll see various organic acids and ATP being exchanged between cells. There's chemical signaling that goes between cells through the plasmodesmata. And one thing that for anybody who's a plant pathologist in here knows that the other thing that can go through here is viruses. Viruses have figured out a way to hijack the machinery that normally functions to transport small RNAs from one cell to the other that's associated with normal plant function, to hijack that machinery to allow nucleic acids to move from one cell to the other to allow the viruses to replicate. Okay, so we've had a reasonable introduction to the sorts of things that I want you to be thinking about as far as plant cell biology in this course. This is going to be reinforced in almost every lecture, because in almost every lecture we're going to talk about stuff at the cellular level and how that dictates what happens at a higher level of organization. Okay, so for Thursday, you are going to read, the assigned readings are in Appendix 1 of the textbook. It's about energy. I also put a document on the website associated with the lecture topic 2 that's called thermodynamics. It's basically my restatement in a way that I think is easier to understand than the textbook of the thermodynamic principles that I want you to understand. You need to understand free energy. You need to understand equilibrium. You need to understand how energy changes dictate the direction that reactions will go and how cells might manipulate that. Okay? So do the readings. Please answer the question that says, what are you having the most trouble with? Because that will help me shape what we talk about next Thursday when we cover this topic. Okay?